Okay, so should we start? Welcome back, everyone, after the break. Our next session, we are very pleased to invite Dr. Souvik Maiti, who's the Senior Principal Scientist at the Institute of Genomics and Integrative Biology at CSIR New Delhi, and he has various other affiliations as well. And he will be speaking about therapeutic applications of gene editing, our experience with the genetic disorder sickle cell anemia, and we hope that this has some applications for GNE myopathy as well. So we're very grateful to have you with us today, Dr. Maiti, and looking forward to your talk, which I think can be around 20 to 25 minutes with
recent work which basically we are doing in our laboratory. I'm a chemist by training, and my collaborator my colleague, Dr. Debojyoti Chakraborty, he is a biologist. So all the work, basically what I am going to share with you, it's a kind of teamwork led by both of us. So in 2017 or 18 onwards, we have started this work. Basically, that was a call from Government of India through my parent organization, CSIR, to start something mission mode project on sickle cell anemia. So there was different aspect of tackling this problem in our country, but we participated with a very basic research idea with that, how we can basically correct sickle cell anemia using our technology, which is called CRISPR-Cas9 technology. So this is a very new technology, was discovered in 2012, and which can basically allow anyone to cut any kind of host DNA, and there are certain process by which you can able to either insert or disrupt or basically change certain genetic material. And for this technology, in 2020, the Nobel Prize was awarded to lady professors, and basically we are excited to start this work for two reasons. One is we wanted to develop a similar technology using CRISPR-Cas, which we will have kind of freedom to operate, so that at the end, if we come out with a successful certain result, it will be very cost effective if we in India, if we want to adopt it. Second thing, definitely it's a kind of problem. I will explain what is the problem in the in this field. When we are talking about any kind of therapeutics, there are two general phenomena, scientific parameters which need to take care. One is definitely activity. That means the technology which you are using or we are using, it should have sufficiently efficacy, as well as they should have a kind of specificity. Specificity, I mean, is that say you are trying to change a particular piece of DNA or some correction in a particular position of a gene. That means the change will happen only that position without basically hampering the other part of the genome. So that is basically kind of challenge still now in the literature. This graph basically kind of give an idea the correlation between the activity or efficacy versus the general specificity. The most characterized Cas9 protein, which is known as SP-Cas9, which also had went to kind of clinical trial, and it's different kind of engineer variant, which basically different for different disease. It is there are lots of clinical trial is going on across the globe. So as you can see that though the SP-Cas9 and their other engineer version of SP-Cas9 has certain amount of efficacy, but when we talk about the specificity, still it is not as optimal as it is required for therapeutic purpose. So we thought that that is kind of basic gap which we can take and kind of do research whether we can improve the specificity with high efficacy. So with this objective, basically Devojyoti, my colleague, has started to characterize different Cas9 of naturally occurring from bacteria. Several dozens of bacteria, bacterial Cas9 protein he was trying to characterize, and accidentally he has landed with a particular Cas9 protein from a bacteria known as Francisella novicita, in short FN Cas9, and has been seen that it has very high specificity when we compare to the known SP-Cas9 protein, which he proposed that can be used for editing mammalian genome itself. But during this experiment, what we have noticed is that the efficacy of this protein is not as great as it can be used for therapeutic purpose. So as a physical chemist, I have chipped in to understand why a particular Cas9 protein has higher specificity, but it has very low efficacy. As it is an enzyme, we started with understanding the kinetics of this enzyme. In this graph below panel, basically we have plotted the cleaved substrate, amount of cleaved substrate as a function of time for different target. I am showing for 3 targets, and the red bars are basically showing for the SP-Cas9, which is basically most characterized and used SP-Cas9, and compared with the protein FN-Cas9, which Devojyoti has described. All these kinetics for different target has shown that the kinetics or cleavage efficiency in terms of the time is very slow than the SP-Cas9, which is most characterized protein in the literature. Then we are interested to understand how this protein is basically binding with its target along with its sgRNA. So we started to measure the binding affinity between these complex Cas9 protein complex along with its target. So when it is a basically we are targeting to a particular target, we have the 100% complementarity, and we measure the binding affinity and compare again with the SP-Cas9. As you can see that SP-Cas9, it is a binding affinity. In inverse of binding affinity has been shown, Kd, the decision constant, is much lower than the decision constant shown by the FN-Cas9. When we measure the similar binding affinity or decision constant, when the target is not perfect, that means there are mismatches between the RNP complex of sp-Cas9 as well as FN-Cas9, we can see that though the sp-Cas9 complex binds, though the binding affinity is lower, but the FN-Cas9s are not able to bind at all. So it has slow kinetics as well as lower binding affinity, which basically Devo has identified. But one of the interesting phenomena which we have noticed that is that if there is a mismatch or there is a not perfect match, perfect target, FN-Cas9 complex doesn't bind at all. And we basically guessed, it was kind of wise guess, that it may be due to this inability to bind to the imperfect target could be a reason of high specificity, but lower binding affinity of FN-Cas9 could be a reason for the low efficacy. So as I have shown that it binds and it binds or it doesn't bind depending on the perfect versus non-perfect match. We use this technology to come out a diagnostics which we used for sickle cell mutation detection in the field. This we have done for for last 3 years. For last 1.5 years we could not do due to the pandemic, but before that and after that also we have done several field trip to Chhattisgarh, Maharashtra as well as Gujarat, and we have explored this technology to detect sickle cell mutation in populations. But when we talk about basically therapeutics, okay, there are two things we have to keep in mind. One is high efficacy, which I showed that it has sp-Cas9 has high efficacy, and high specificity, which basically FN-Cas9 has. But both the protein has drawback in terms of for sp-Cas9 low specificity as well as for FN-Cas9 is a low efficacy. So if we go back and try to understand the textbook chemical kinetics of this kind of enzymes, so there are 3 parameters basically we can define. One is the dissection of the binding affinity, which is nothing but the ratio of K on and K off. K on is basically the reaction rate of the forward reactions, and K off basically backward reaction rate. So that ratio basically we measured initially. But if we basically try to understand specificity in terms of these parameters, when the K off divided by K, ratio of K off and K cat is less than 1, we call it is a sticky equilibrium. In this case, the window of specificity is in the lower hand side. Whereas when the K off by K cat is greater than 1, we call it is a rapid equilibrium from enzymes, and we basically end up with high specificity, which means that if we can increase the K on, which is basically forward reaction rate constant for the protein we are dealing with, we may achieve the higher specificity. So whether it is true or not, to understand that, we did kind of single molecule kinetic study. What we understood that sp-Cas9 as well as FN-Cas9 both occupy in the same time when they are approaching to the target. Whereas when they are going out of the target, FN-Cas9 takes less time to go out than the sp-Cas9. In a quantitative way, if we just dissect K on and K off, the binding affinity ratio, we see that the K on, the forward rate constant for the both protein sp-Cas9 as well as FN-Cas9 are more or less same. Whereas if we measure the K off and quantify, the K off, which is basically dissociation rate constant, is much higher than for the FN-Cas9 than the sp-Cas9. And we realized that this is the this is the reason for which the sp-Cas9 has high efficacy rather low specificity, whereas the FN-Cas9, which Devo has discovered, has high specificity but low efficacy. So this is the summarized. This is the table which summarize all the observations which we have made, and the main reason of low specificity for sp-Cas9 is nothing but lower forward rate constant, higher forward rate constant than the in a chemical kinetics rate constant, enzymatic kinetics rate constant. Whereas in case of FN-Cas9, K off is higher than K cat. That's why it is showing high specificity. So this led us to make certain kind of engineering of this system such a way that we can increase K on, which is forward rate constant, so that we can increase the binding affinity between the RNP complex and the target, so that we can achieve the high efficacy without hampering the specificity of the protein. There are three ways basically you can do that. One, you can change the length of the cRNA, as well as you can place some kind of chemical modified oligonucleotide or bases to increase the binding affinity by increasing the K on between the RNP complex and the target DNA, or you can basically engineer the protein so that the K on of the forward reaction rate constant is increasing without hampering the backward reaction rate constant. So we basically took all three approaches, like changing the length of the cRNA, as you can see, notice here that there is optimal sgRNA 21. That means length is up to 21 mark oligonucleotide. We have the best efficacy. We are using also, there are modified oligonucleotide we call lock nucleic acid to increase the K on of the system. Rather, we also are interested to engineer the protein so that we can increase the binding affinity. So for that reason, we have collaborated with one of the pioneer in this field, who basically solved most of the crystal structure of the most of the Cas9 protein, Osamu Nureki, and we started engineering the protein with the ambition that we can increase the forward reaction constant between the complex as well as the target DNA. We came out almost 50 odd engineered protein, and we tried to characterize them. And here we see again measure the percentage of cleavage as a function of time, because that was the limitation for the natural FN-Cas9. As you can see, there are a couple of engineered protein, at least EN1, EN15, and EN31. These three engineered protein has very, very high efficacy when, again, if we compare with the SP-Cas9. We also measured its specificity, whether it can retain the specificity or not. We have measured, and they have sufficiently, their specificity has not changed. They are retaining the high specificity. This is basically some biological data to show you. Most of the work, again, done by Devu's lab, but just to convince you is that, the engineered, FN-Cas9, this is EN1. In almost all the cases, the target we have, selected, it has very high efficacy with respect to either, engineered SP-Cas9 or natural SP-Cas9. So these are basically kind of proof of concept which we have done in our lab. With this, we got an opportunity, in a Sickle Cell Day. Our director were presenting this data in a similar way that we are organizing here. There happened to be present by Secretary of Minister of Tribal Affairs, and we got an invitation from Minister of Tribal Affairs whether we can start kind of clinical trial for sickle cell anemia using our developed technology. So most of you may know that is a single base mutation which basically causes sickle cell anemia disease. So it is easy to handle for us because only one base we have to change. And this is the scenario of of Indian load of sickle cell anemia patient. This is per 1 million 10 lakhs for 10,000 100,000 patient per patient. Almost more than 90% are the deep color which has been shown in the map. So it's a huge load. So what we have proposed in the clinical trial or preclinical trial proposal is that there are two approaches we are going to take. one is ex vivo approach, where we want to take out the patient stem cells which is defective, and then in laboratory we are going to correct. So for that we need again kind of certain delivery technology, which basically Orco, who is the earlier speaker, who is helping us, as well as we also aim to try with in vivo correction, where complex with AAV vector, which basically Orgo has explained few minutes back, so that we can inject that within the patient and it can be corrected in vivo. So both the approach we have proposed in our clinical trial. It is not that we are doing the clinical trial. There is a huge group. Basically, we are the technology platform provider, platform technology provider. The main clinical partner are coming from AIMS Delhi, Dr. Rishi Dhawan, Dr. Ganesh, Dr. Tulika Seth, and Dr. Mahapatra. They are basically clinicians who are going to do the clinical trial. We have different partners, like Orco is helping us to develop AAV vectors for delivery. From NIH from Mumbai, they are going to study the immunological effect of this Cas9 protein when we inject into initially in animals or then patient. Then we have certain collaborator from Sickle Cell Anemia Institute from Chhattisgarh and other part of the country. Most importantly, we are having a lot of good mentors from globe, those who are part of different stage of clinical trial for particularly sickle cell anemia. We have Drew Weissman from University of Pennsylvania. We are having good support from Dr. Akshay Sharma, who is actually parallelly running a clinical trial for sickle cell anemia in US. We have also doctors from US who are basically very much into different kind of clinical trial for hematological disorder, like Hema Rangarajan, like Laxman. Okay, all of them are kind of extended their support, and we have placed our MOU and other necessary understanding between all these mentors as well as the group member within this clinical trial team, and hopefully we'll start this work very soon with the ambition, because we are very much hopeful already. Very encouraging information or news are coming that CRISPR treatment has been successful in couple of places, including US, and that even for blood disorder like sickle cell anemia and thalassemia, and with the dream that one day our technology will be used in our country, where our doctor will be happy to declare that yes, using this technology our patients are getting cured. With this, let me thank all of you for your time to spare, as well as my group member, my collaborator Devajyoti Chakraborty, as well as till so far we are got good funding from Council of Scientific and Industrial Research. This is my parent organization. With that, I thank you all, and I will be happy to answer any of your queries you have. Thank you very much.
Thank you very much, Dr. Maiti. That was a very comprehensive and very easy to understand presentation. Thank you for making it so well. We have one hand raised. We have one question in the chat box as well. So maybe we go with the hand raised first. Shri Darshini.
Yeah, it's very nice work you have done. I read this paper, I mean, before also. I have a question that the major concern about the Cas9 is, of course, the off-target, and the Xiang lab developed SP Cas9 variant, that D10A exported to alanine, I guess, in the 10 position.
Yeah.
So to reduce the off-target, and how, have you compare your FN Cas9 with how in case of efficacy, because it target only one strand to reduce the target and using two Cas9 at the same time to increase the efficiency or on target. So have you compared both? Or
Yeah, I have shown one slide. We are taking 3 different commercially available, high fidelity SP-Cas9 as well as other 2, and comparing with our own engineered FN-Cas9 protein in 2 different aspects. One is definitely efficacy as well as the specificity. And I'm glad to share that, engineer FN-Cas9 is as good as the best version available and at this point in the literature. So we are further characterizing, because this is just preliminary data. We have to characterize genome wide of target, which we have already done for few cases, but we have to keep on doing more and more to confirm that the observation which we have in our hand right now is indeed correct. But first few experiment definitely showed that the protein which we have generated as good as it is reported in literature. Not only that, just to give you a more better confidence. So after sharing this data with many of the leaders of this field, we have shared our own proteins to compare along with their proteins, and most of the cases we are getting very encouraging responses that this protein is working as good as either they have developed or other protein those are known to be good in the literature.
Okay, so one question, another question is how big is the FN-Cas9? I don't know whether I missed it in the slide or
Yeah, FN-Cas9 it's little bit bigger than the SP-Cas9. SP-Cas9 is 1100, close to 1100 amino acids, but FN-Cas9 is 1584 amino acids. So what we are trying to do the next is that in the proteins we are trying to identify that which are the portion is not necessary for its editing as well as other parameters. We are trying to engineer and get rid of and trying to make it as small as possible.
Yes, because packaging in AAV will be very difficult if it's a large protein.
We are taking 2 alternatives. One is definitely we are trying to reduce the size of the protein, as well as we are trying to take advantage of mRNA based delivery. So where the size would not be that critical.
Okay, thank you so much.
If I can add one thing, so on the AAV side, we are developing the dual vectors. So my own past work has been on dual vectors, and these dual vectors that I have developed in the past have already been licensed for human trials. So we are going to take a similar approach to accommodate the full-length Cas9.
So we only have a few more minutes, Dr. Sudha Bhattacharya, and then Dr. Maiti, if you can answer the questions in the chat later on, that would be
certainly I'll do that.
Souvik, great talk and very encouraging for all of us that you're making such headway. My question is that ultimately you will have to use your Cas9 for editing the sickle cell mutation. So are you coupling it with a base editor also?
So we are developing also base editor, keeping the FN-Cas9 backbone. So this is under research mode. We are basically having couple of version, but we are characterizing. Yes, we are into developing to base editors.
So will you be having a whole range of base editors or only 1 or 2 specific?
Our ambition come out with one after another base editors. We have started A to G. So at this moment, but definitely in a long run we will come out, because one of the advantage of this that we have patented, and almost 1.5 years it is there. So we are hopeful that we will get it. The indications, whatever the fighting is happening, we will get it. So once if we have our parent patent, that means we can develop all kind of Cas9 based next generation of genome editing tools around this. So that basically one of the critical, unless until if we do not have our own freedom to operate, developing actually doesn't help too much other than satisfying the basic research interest. So we are basically, waiting that whether the patent will be granted to us, then only we will take up in a larger scale to develop all kind of, next generation base editing, like, base editors or prime editors around this technology. We will take up.
Thank you.
Are there any other questions? Okay, otherwise I think let's wrap up the session. And thank you very much, Dr. Maiti. It was really a pleasure to listen to you, and we hope to collaborate with you as well in the future. So thank you very much.
Thanks a lot.
And now I will hand over to Dr. Sudha Bhattacharya for the session that I think a lot of people are waiting